Welcome everybody to the UW-Madison Biotechnology Center. I'm Tom Zinnan. I work here at the BioTrek Center. I also work for Wisconsin 4-H, and today we're going to extract DNA. Liz, would you like to say hello?
Hi, I'm Liz Jesse. I also work at the Biotechnology Center and for UW Extension 4-H. Today we are going to extract DNA in two different locations, which is kind of great because you're at home and I'm at home and Tom is in the lab. So we're going to kind of show a couple different ways to do this. Now, you maybe have extracted DNA before. You maybe in school you did it with a cheek swab, or maybe you did it with strawberries or kiwi. There's a lot of different things that you can extract DNA from. But in our lab at BioTrek, we extract it from something that is very near and dear to us in Wisconsin. And I do a lot of biking, and I bike out into the countryside a lot in the summer. So Tom, maybe do you have something in front of you that's going to help us figure out what we're going to extract DNA from today?
Well, we've got if we can open up what we have.
Okay.
This stuff, and if you want to show them that, I'll go grab the bag of the other stuff.
Yeah, absolutely. So I'll see if I can get close enough to the camera so you can see what we are going to extract it from today. We work with K through 12 with K through 12 students, and we get a lot of different responses for what we think this is. Eventually, students kind of work their way to it is a type of grain. So we talk about the different types of grains that are in Wisconsin, and we talk about rye, and we talk about barley, and we talk about, Tom, what do you have in front of you?
This is some 3-year-old wheat, heads of wheat, and the heads of wheat produce the kernels, and the kernels are where we get wheat germ from. So this is what this looks like. This is from Broadhead 2018.
All right, so we are going to be using wheat germ. Now you can buy wheat germ at most grocery stores. And if you go down the flour aisle, you'll see there are specialty flours, and there you probably will see a little bag of wheat germ. Often it is Bob's Red Mill, and that is what we use. Tom maybe has a bag in front of you that he can show. Excellent, go ahead, Tom.
So this is raw wheat germ. It'll be refrigerated or frozen. We keep it in the freezer. Avoid the toasted stuff that comes in a jar, because for our purposes it doesn't yield the kind of DNA glop that we're looking for.
Excellent. All right, and I'm going to be using a conical tube, a graduated conical tube today, which is what we use in the lab. And this is all summer. Well, actually, all since pandemic, we have been sending kits to students that have these graduated tubes. But if you're at home, you can use something else that you may find in your medicine cabinet. Tom is going to be using that. So Tom, what are you going to be using?
Well, here's one with the label on it. And if you take the label off, you've got this nice little pill jar. There are many sizes of pill jars. This one works pretty good. And it's already got the wheat germ in it. So you can see about how much wheat germ you need to put in there. We're shooting for a gram, and I think if you put in a heaping quarter teaspoon of raw wheat germ, that'll be pretty close.
Perfect. Okay, sounds good. All right, so and then we kind of talk about then where DNA is found in the cell. Okay, and we say that if you had this wheat germ and if you put it under a microscope, you would probably see boxes. And anytime biologists see boxes, that immediately indicates cells to them. So then we would talk about where the DNA is found in the cell. We would eventually get to that it is found in the nucleus. But here's the problem. We have the DNA stuck in the nucleus right now. So our first task in the extraction is to swell that nucleus, all right? So it gets to the point where the nuclear membrane is just about ready to pop. So we're going to use water. Now, if you have a little conical tube like me, all right, and it can be any kind of water. It can be tap water. It can be anything. It's fine. I'm going to fill it to about 25 milliliters. I'm a little over, but that's okay. Tom, how much are you filling yours?
Oh, about 1/3, 1/4, something like that, because we're going to have plenty of space to add something later on, the alcohol that we'll be adding later on.
Great. And then you're going to close your top and you're going to start shaking it. Okay. So you can shake it pretty aggressively right now because the DNA is stuck in that nuclear membrane, which is great because it's semi-permeable membrane. So the water can get in, but everything else can't get out. So go ahead and shake it. Shake it. Use your favorite shaking song. Tom has some favorites, I know.
It's a bad joke alert. We use the shaking song to get across the idea DNA is a lot like an alphabet. just like with the English alphabet where we spell words by ordering the 26 letters in different orders to give different words with different meanings. DNA is a lot like that. Instead of having 26 letters, there's only 4, but different orders give different genes with different functions. And if you make small changes in the order, either in the spelling of words or in the making of genes, you can get a significant difference. For example, one of my favorite shaking songs is Shake, 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 Shake Your Booty. And if you take the word booty and switch it around, switch the B and the T around, you get shake, 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 shake your tubie. That's a bad joke that illustrates how important slight changes in letters sequence can be.
Nice. All right, so at this point, after you've sung whatever tune you feel like singing, you can stop shaking, you can pop your top, you can give it a nice little waft, proper technique for sniffing is not sticking your nose right in it. People all have, our olfactory senses are different, right? So I smell green grass, I smell green bananas, or you may smell something different. Stick your finger in it, you can feel that it's a little bit slimy. And you can also, if you get close, this looks very different from when we originally put the water in, right? Looks really cloudy, it looks like milky. So we have these swelled cells, these swelled nucleuses right now, nuclei. And to get in there, we need to pop them. So we are going to use a chemical needle. We're going to use a surfactant, that is Dawn dish soap, all right? In the lab, we put them in these nice little 1.5 mil centrifuge tubes, because this is going to be eventually where we're going to put the DNA that we extract out. So you'll need another kind of small container, but it's about probably, what do we decide, Tom, about 1/4 of a teaspoon of soap?
A teaspoon is about 5 mils. This is 1 mil. So if you do a 1/4 teaspoon, you'll be in the right neighborhood.
Good. All right. So yeah, we can let it sit there for a little bit. It's okay. You're not going to get all of it in. As long as you get a little bit in, you're going to get it done, because we've been washing our hands a lot for the last year, right? Because surfactants are really good at breaking up things, breaking up dirt, breaking up fat, breaking up particles. That's going to break up that nuclear membrane. All right, now we need to be a little more gentle, because DNA is a little more fragile once it gets out of the nuclear membrane. No more shaking. You're going to rock it, okay? And when we do this in the lab, we take a couple seconds to discuss what we know about DNA, and we kind of use an analogy. So our analogy is, Tom, you want to talk about our analogy?
Well, DNA is a lot like an alphabet, but it's also a lot like a recipe card. So if I gave you the choice between having a plate full of cookies on one hand or the recipe card for cookies on the other hand, which would you take and why? And since this is college, it's nice to be able to give the pros and cons of each choice. So have a little conversation with yourself or with your friends. Think about what's the good points and the bad points of taking the recipe card, and what are the good points and the bad points of taking the cookies. I always come down in favor of the cookies, but that's just me.
I always come down in the favor of the recipe card, but we're different.
Person. So the cool thing about this is recipe cards are a lot like DNA and vice versa. You can store them, you can retrieve them, you can edit them, you can copy them, you can share them, you can send them over space and time, and you can do all that stuff with DNA. And so DNA and recipe cards are both a form of recorded information. And that's one reason why so many people in this Genetics Biotech Center building here work on our friend DNA, because where's most of the genetic information recorded? On DNA.
All right, so we can stop shaking at this point, and I'm gonna get my camera close enough. You can see it looks a lot, it's a suspension right now. So it's kind of like Italian dressing. Everything is floating in there because everything that was trapped in the nucleus is now out into the bottle. But we don't want everything, right? We just want the DNA. So the next step is called a precipitation. We're going to precipitate the DNA up out of the solution. And we're going to use that, we're going to do that using alcohol. Now, if you're doing this at home, you want your alcohol to be, Tom has 70% right there. The higher, the better. There's 90, there's 91% isopropyl alcohol. The higher the alcohol content, the better. And you want it to be extremely cold. So keep it in the freezer before you use it. and then take it out right before you're ready to use it. Mine is high enough that in the lab that I don't have to keep it in the freezer. And you're going to fill it. Be careful. Think water slide, not cannonball. Remember, we're trying to create 2 separate layers with the alcohol on top and everything else that we don't want underneath. And the DNA will come up into that alcohol layer. So pour it in as much as you can get in there. All right, to the very top. Okay. So what you should see is a layer, and then you'll see it's a little cloudy things floating in there. Kind of looks like a jellyfish, kind of looks like snot, looks like spit, a lot of different fun stuff. But what that is DNA glop. We need to get it out. So how you get it out is we have these wooden Q-tips, these long 6-inch wooden Q-tips. Wood works really well because DNA will stick right onto it. So you're going to go in kind of like you're going fishing, and you're going to stay just in that alcohol layer, and you're going to swirl it around. And you don't go into the layer underneath because that's the stuff we don't want. And when you swirl it and you twist it, you should come out with a fantastic looking glop. Okay.
I don't have a long wood stick. I've just used a
straw, drinking straw. Great.
Good.
Or a knife or a fork. I think a lot of things would work depending on your container. And then you're going to want to slide it into something where you can keep it. All right. So I have this little, in the lab, we use these little 1.5 mil tubes, which Tom has and I have. It could be anything that you could then close up and keep it now to be around for a while. So now you have a sample of DNA that you extracted yourself at home. All right, anything to add, Tom, before we sign off?
Yeah, like you mentioned, this is, we call it DNA glop because it's not highly purified DNA, but it reflects how researchers go to get their DNA. You start with a cell, you break it open, you precipitate the DNA with some alcohol, and then you either spool it up or get it another way.
All right, thanks for joining us.
Thank you.